Hi, okay, so this is endocrine. This is the last part, so part 3 of 3, and we're going to talk about the pancreas, diabetes, calcium homeostasis, the parathyroid gland, things like that. So these are your abbreviation reference. So these are all the abbreviations, just in case I forget to put them in there, or it's just kind of something to review, to look at. Same thing here. These are all your endocrine glands, hormones, classifications, actions. This is another way of looking at the hormones that we've covered so far. So we've covered the hypothalamus and the anterior pituitary, so the HP axis. So HP, hypothalamus, pituitary axis. And we'll come back to those when we talk about the reproductive hormones. But right now, we're not going to cover hormones that are kind of directed by this HP axis. So we're going to look at the parathyroid gland, which is releasing parathyroid hormone. It kind of has its own sensors. It doesn't move through the through that HP axis. Same with the kidney and the pancreas and the release of insulin, glucagon. Okay, so let's start with the pancreas. So the endocrine pancreas is what we're talking about. 99% of the pancreas function is exocrine. So if we look down here, this is the pancreas. It's kind of behind your stomach. It's always in all these pictures. It's always nicely formed and shaped. Usually, if you look at it in a cadaver or in a like an animal or something, it's just this mushy mess that's kind of behind the stomach. But it is organized. So maybe I've just had bad experiences, but it is organized, and so you can see the exocrine function. You have the production of these enzymes, then it moves through the pancreatic duct to the joins up with the common bile duct, and then is excreted into or secreted into the stomach. So in this case, we're talking about these little islets of Langerhans. So these little islet cells here, which is a bunch of different cells in the islets of Langerhans. And those are going to release into the blood. Okay, so here you have the bloodstream up here. So you have a bunch of little capillaries down here, and those are going to feed into those capillaries, and then it's going to be released into the blood. And it has to, because that's what hormones do. Hormones are released into the blood. So the pancreas releases hormones glucagon, insulin, and somatostatin. But into the bloodstream. We're going to talk a lot about glucagon, insulin. Mainly, we're going to talk about insulin, but these are all important. So these hormones are released from these little islets. Yeah, the islets of Langerhans. So if we look at the islets of Langerhans, so there are about 1 million islets within each containing about 2,500 cells. So even though it's only 1% of the pancreas function is this endocrine function, there are 1 million of these islets, and they each contain 2,500. So this is kind of a cartoon representation. Obviously, there aren't 2,500 of these, but it kind of shows how these different cells are arranged. So the main cells, there are more than just 3, but we're going to talk about 3. Include alpha cells, beta cells, and delta cells. So those are the big ones. And actually, if you let's see, if you do the math here, you get about 95%. Okay, are these cells alone? But then there are other cells, like there are F cells and some others that we're not gonna talk about. So the alpha cells, 20% of the cells of the islets, or the islets of Langerhans, are alpha cells, and you can kind of see here where they're kind of around the outer part of the islets. And then you can see insulin is beta cells produce insulin, and that's 65%. So you can see kind of these mauve, yellowish kind of colored ones here are the beta cells. So you can see that it's primarily over half beta cells. And they're releasing insulin. So then you have 10%, which are the somatostatin cells. So somatostatin is kind of influence. Somatostatin can actually inhibit both beta cells and alpha cells, but they kind of have this paracrine function within the islets of Langerhans, and they kind of, they're kind of a control mechanism to prevent too much release of either one of these. All right. So yeah, just said alpha cells are mostly around the outer rim. Delta cells, somatostatin serve a paracrine role within the pancreas. So we've seen a lot of stuff that somatostatin has done in our various sections that we've talked about. Most of it is kind of controlling things, controlling growth hormone, controlling digestive hormones. All right, so reducing. All right, so beta cells release insulin when they detect high blood glucose. So this is a big factor that is causing the release of insulin. There are other gut-derived hormones that will also cause the release of insulin. There is also the smell of food can cause the release of insulin. But the main thing that we care about, well, we care about all of them, but the main thing that actually causes this is this interaction between blood glucose and the release of insulin. So that's a I guess the primary control mechanism for releasing insulin is just high blood glucose. All right, so alpha cells release glucagon when they detect low blood glucose, such as between meals, and then delta cells produce somatostatin, which extends the time food is in the stomach, blocks insulin and glucagon release to give time for cells to absorb nutrients. So they're kind of a modulating. They play kind of a modulating role. So you don't want blood. So for instance, they can be, if blood glucose levels get too low, then they can block the release of insulin. But then they can also block the release of glucagon when that's overactive too. Okay, so they're just different control mechanisms to modulate that. Okay, or to control that. So insulin, beta cells, like we said, release insulin when they detect high blood glucose levels or when triggered by gut hormones. So it is some interesting and kind of useful facts about insulin. Insulin is the first hormone isolated from animal sources in a form that could be administered therapeutically to humans. So this started out as just grinding up pancreas. So there was something in the pancreas that was able to lower blood glucose because I want to say in children, but in type 1 diabetics, because it started out that, nobody really knew what was going on, except that some people would develop this high blood glucose with sweet urine, and then they would have a lot of other problems, and then die at a very young age. So yeah, it was first isolated, I believe, in a dog, and so from this pancreas, and then the pancreas juices could be injected, and it would actually lower blood glucose. So they knew it was something in the pancreas, and then they went on to isolate the actual peptide that was causing this, and that was insulin. So now insulin is used to be produced in, I think, pig cells, pig pancreas. They would just harvest those, grind them up, isolate the insulin, and then use that. But now it's produced using recombinant DNA in bacterial cells. So you can get it a lot quicker. You don't have to use pigs. Bacteria are a lot smaller, and they just kind of pump it out. Okay, and when they pump it out, though, it's not in the final form that that DNA is. So, well, I mean, it takes several steps to actually produce the insulin. So insulin is actually two different chains, and if we look down here, it's really this, and we just ignore all this. Okay. So cleavage enzymes or restriction enzymes will cut that off, and this right here is insulin. Okay, just those two chains connected together by disulfide bonds, that's insulin. But in order to make insulin, you can't just make two chains and then hope they match up. You have to actually go through the whole process of transcription in the nucleus to make the mRNA, and then the mRNA will make this long chain that includes a signal peptide. Then you cleave off the signal peptides. The signal peptide will tell it that it has to go to like the ribosomes, and then you cleave that off, and then you have this product left over. So this would be a pre-pro hormone. I don't expect you to know all of the steps, pre-pro hormone to pro hormone, but I do think it's important to know that that it comes from this longer chain, and then this other part of this chain has to be cut off. So this is, or has to be cut off. So this is the connecting peptides. We can call it that because these are our two chains, and this is kind of connecting those peptides. So the connecting peptide, or the C peptide, that ultimately has to be cleaved off. Okay, so that's where we cut that, or we don't do it, your body does it, with restriction enzyme or proteases. We'll cut it, and then it will leave the final product. Okay. Now, the reason we want to know about this and we talk about this is because if you have a patient who has, I don't know, let's say that for some reason their blood glucose is running low, low, low all the time, and maybe you suspect that their beta cells are actually functioning. So you're normally in a type 1 diabetic, their beta cells are just gone. So the immune system wipes them out. But but it's possible, or if you have a new patient and you want to see, but if you have a patient who's actually taking insulin injections, it's going to be hard to go in, take their blood, and say, okay, I wonder if they have insulin in their blood from these beta cells producing the insulin. And of course they do if they're taking insulin injections. Okay, so how do we tell if the beta cells are producing insulin versus is this just insulin that was injected? Okay, so the way to do that would be to look at the C peptide, because when the patient injects insulin, they're not going to inject C peptide. So the only way that C peptide is going to be present is if it was made in a beta cell. Okay, so that's because that's where insulin is made. So that's one use of this C peptide, is to monitor or measure production of insulin by the beta cells. Okay, so I hope that makes sense. Okay, so yeah, so it works even when that patient is still taking exogenous insulin. We can still see if their beta cells are functioning. All right. So factors affecting insulin secretion. So these are stimulatory factors that have been found to increase insulin secretion, and so that's just something that has been tested. So all of these have been found to do that. The most important of these for both stimulation and inhibition is glucose concentration. So for stimulation, increased glucose concentration, and for inhibition, decreased glucose concentration or decreased blood glucose. So it's not saying that these others aren't important. It's just saying that this is the primary driver for it. But other things, increased amino acids, because if we think, and we're going to talk about what insulin does, if we look at what insulin does, it's trying to store energy, or it's trying to store nutrients, those kinds of things. So so increased amino acid concentration, well, amino acids, we want those for later so they can be turned into proteins. Fatty acids can be turned into triglycerides, just adipose tissue in fat cells. So we want to store that stuff. So those also, so the increase in amino acids, increased fatty acids, those can also stimulate insulin, but that's not the primary driver. In fact, so oral glucose increases several gastrointestinal factors. So we have this here. This is an example. It's not one that you have to necessarily memorize, but it's an example of when you have a meal. So we're talking about stimulating insulin release. When you have a meal, when food moves into the small intestine, then there's going to be sugars, glucose there, and that will trigger the release of hormones from the small intestine to the pancreas saying, hey, we have sugar arriving, and so it increases the insulin release from the pancreas. So glucose-dependent insulinotropic peptide is a peptide that will then, it's a peptide hormone that will then go and let the pancreas know that there's glucose in the stomach. Okay, but and kind of an important point of this is that if you provide, so this is why oral glucose is a more effective stimulator of insulin release than IV glucose. So if you give somebody IV glucose, okay, and it just goes, that's supposed to be a needle in somebody's arm, and it just goes right into the bloodstream without going through the intestine. So this is supposed to be a stomach with the intestine. So this would be an oral administration. Well, that's going to cause the release of these hormones that can then go tell the pancreas to release insulin. Okay, if you just give it in the blood, you don't have this bonus stimulation of insulin. So you can kind of see why oral administration of glucose is going to more effectively stimulate insulin release than IV glucose. Okay, so hopefully that makes sense. If you eat it, you're going to stimulate your insulin more than if you inject it, okay, because the gut's paying attention. All right, so there are adrenergic receptors on beta cells that will inhibit insulin release as well. So if you have a fight or flight kind of situation, okay, so kind of think about this. If you have a fight or flight situation, do you want, okay, so there are your choices. You can either have glucose in the blood out there ready in case your muscles need it or something needs it, probably muscles if it's fight or flight, or do you want that insulin to be, or that glucose to be taken up into cells and stored? Okay, so we have to think about this when we think about the function of insulin. So this is what insulin, the overarching function of insulin, is to take glucose out of the blood and move it into cells for storage. Okay, so yeah, if you have, maybe you already answered the question, but if you have a fight or flight situation, if you're being, chased by a walrus, a dog, or a bear, I don't know why I wanted to say walrus, that's weird. Maybe if you're in the water, but if you are being chased by something, okay, extreme example, then you want to have those nutrients available. You want to have this glucose available so you can use it. You don't want it to be stored, and since insulin is storing it, then that's why you would have adrenergic agonists that are going to inhibit it. Same thing with fasting. If you're not eating, then you don't necessarily want to store that energy. You want to have it available. Same with exercise. You want to have it available. Okay. All right, so regulation of insulin secretion. So we know that insulin is secreted with high blood glucose. Okay, so we can just kind of say that that's been discovered, and then these very smart scientists said, well, how does that work? How is it that glucose is being is stimulating the release of insulin? And it's an important thing to know, because when we start getting into like type 2 diabetes, then we kind of want to know how this mechanism is taking place, because a lot of times beta cells will become insensitive to glucose and they won't work properly. So let's just look at kind of we're going to simplify the simplified version. So the simplified version is over here. There are a lot more steps that take place to get to this point. But we're just going to kind of look at the overarching way that glucose stimulates insulin release. First of all, when you have high glucose, okay, so lots of, I don't want to make it like a six-sided, but either way, so glucose is found in high concentrations, then it is going to activate glucose transporters to move it into the cell. So just like in the kidney, when you have glucose, you have transporters that are causing the reabsorption back into the body, And the more insulin, the more active these glucose transporters are. Of course, we know that they can get saturated at a certain point, but within a range, the more glucose means more transport of glucose into the cell. So if we look at in these beta cells, okay, so we're not in the kidneys right now, we're in the pancreas of these beta cells, and we say, okay, well, glucose moved into the cell. What happens to the glucose? Well, through a number of steps, we have the production of something called glucose 6-phosphate, which is also in the glycolysis pathway and the production of ATP. But anyway, it's oxidation occurs from this, and you have the production of ATP. So when glucose moves into the cell, okay, we have the production very quickly of more ATP. Okay, so this is in the beta cells, so it's not necessarily concerned about, you know, muscle contractions or anything like that. This is a glucose sensor mechanism. That's the whole purpose of it. So when the glucose moves in, it makes ATP. Well, there's this little channel over here, a potassium channel, that is sensitive to ATP. When there is a lot of ATP, this channel will close. Okay. Now, another thing we know, and it's a potassium channel, is that potassium is constantly kind of leaking out of the cell. And we also know that potassium is positively charged, right? So we know that the inside of the cell is negative, and the outside of the cell is comparatively positive, okay, so that's polarized. So if you stop this channel from allowing potassium out, then what you have is potassium building up inside. And suddenly you have depolarization. Okay? So that additional potassium building up inside the cell will cause depolarization of this cell. Now, it's not like an action potential, so it's not the same type of depolarization. I mean, it's the same depolarization as depolarization. It goes from being, I wish I knew the numbers here, but let's say -50 to reaching a threshold of -20 or something like that. So that is a depolarization event. That depolarization event will then cause calcium channels to respond, allow calcium in, and cause exocytosis. All right? So remember the calcium, I don't know if you remember, like the snares that pull those vesicles down and then release. Okay, so that's what we saw in neurotransmitter in neurons, but here it's releasing insulin. So it's kind of the same mechanism. Your body's really good. It's like, hey, this knows how to do this. Let's use it. So calcium binding to, like snare proteins or whatever, can move a vesicle down and release the contents of the vesicle. So we have this little depolarization event that happens, which causes that calcium to move in and the release of insulin. So the point of this is, well, a couple of things. So a couple of things are good about knowing this. One is that we know that there are these mechanisms that can be used not just in neurons or muscle, but the same mechanism, this depolarization mechanism, can occur in other cells as well. So it's good so we don't say, oh, I thought depolarization only happened in those. No, it's just a feature of cells that of the body, of physiology, that can be done. And it's useful. The other thing is that we have these glucose transporters that are sensing the glucose moving in. Okay, so we do have this mechanism that will then cause the release of insulin. Okay, and so we kind of know how insulin is monitored. Okay. All right. It's also a good review for the neuro stuff, because you get to think about potassium again. Okay, so insulin mechanism of action. So how does it happen? So we have insulin. Insulin has been released. Now where's it going to go? What's it going to do? It's going to bind to receptors on various cells, usually fat cells, muscle cells, and liver cells. But it binds to these receptors, and these are tyrosine kinase receptors. They kind of occur as a dimer or two of them together. Okay, that's some cell biology for you, but it's made up of an alpha subunit, beta subunit, and then you've got your little disulfide bonds holding everything together. Okay, so that's just the structure of an insulin receptor. What we're interested in is that the insulin will bind to a receptor. Okay? And when they bind, they will cause these receptors to have an internal mechanism, kind of like we saw with G protein coupled receptors, but they'll be like the second messenger mechanism, and where they phosphorylate things, and then they will activate signaling proteins that will then go. Those signaling proteins are kind of that increased response inside the cell, and it'll cause all of these things to happen. Okay, so insulin binds and causes a response inside the cell. In this case, we can see what insulin is doing. Okay, so in this case, we're saying it's fat synthesis, and by fat synthesis, I mean taking fatty acids, okay, and then binding them to a glycerol to make triglycerides. Okay, so that's your fat. So you're making these fats, and that's how fats are stored, is through these triglycerides. So fat synthesis, storage of energy. Okay, so that's the theme here. We have amino acids that are being brought in. Okay, so amino acids are coming in, and so protein synthesis. We have growth and gene expression. Growth and expression is activated also, so affected by insulin. Okay, so that's kind of a, when we talk about growth, maybe we're talking about muscle growth during but gene expression overall is taking place. Okay, so things are happening inside the cell. Now the other thing that is going to happen, so consider this down here as storage of energy, but over here we have glucose that is going to start moving in through glucose transporters. These are the GLUT4s. So the glucose transporters are going to allow glucose to move into the cell, and it is also going to be stored. It's going to be stored as glycogen. Okay, so it's just going to be put in these long chains. All right, so the receptor activates or inhibits proteins to produce its effect. Okay. So we have the effect. We're gonna kind of get into some of the other details of exactly what's going on with that storage. But if we look at the receptor, and here's the receptor, it just did its job. It turned on these mechanisms inside the cell. It caused glucose transporters to be distributed into the membrane. And so what happens to the receptor? Maybe you just say, well, nothing, it just sits there, but that's not the case. So after activation, the receptors are actually internalized. Okay, so they are internalized and will either be degraded, or then they will move back out and be placed in the membrane again. Okay, but they are downregulated after activation. So this is a function to prevent your blood glucose from becoming too low. So overactivation of the insulin receptors. So if you have a little bit of insulin, or if you have insulin that's released after a meal, and so it's running around in your blood, you could kind of see a situation where there's this insulin, and if these receptors are just out there, and the insulin isn't broken down very, very rapidly, then it's just going to keep activating these receptors, and all the glucose is just going to move into the cells, and your blood glucose is going to drop too much. So if you have this delay, these receptors that are internalized and then either broken down or put back in, then we have kind of a mechanism to prevent glucose from going too low. So the insulin downregulates its own receptor. So inappropriate downregulation of receptors is partially responsible for insulin resistance in obesity and type 2 diabetes mellitus. So type 2 diabetes mellitus is a situation, at least initially, where you have insulin release from the pancreas, but when it goes around to activate this receptor may not be there. Okay, so it may be downregulated and it may stay downregulated. Okay, so why that happens, nobody knows, but that is one mechanism for type 2 diabetes. Another is that some of these internal mechanisms aren't working, particularly the one that puts this transporter into the membrane. Okay, we're kind of jumping ahead of ourselves here. Okay, so review: insulin binds, causes responses that overall are going to cause the storage of energy and movement of glucose into the cell, and then the insulin receptor is internalized, and then it's either broken down or it's recycled and moved back out. But it's downregulated by the binding of insulin. Okay, so one of the things that was not very well shown on the previous picture is how is glucose moved into cells based on insulin binding. Okay, so insulin binds to and activates those receptors that we call that dimerized tyrosine kinase receptor. So it binds to and activates those receptors, and those intracellular signals that we saw, and there are a bunch of them, are generated that result. So one of the things that it does is the insertion of a glucose transporter. Okay, so these glucose transporters are kind of sitting down here in these little, what we would call maybe synaptosomes, but they're just kind of sitting down there saying, you know, if you need me, let me know. And this is phospholipids. It's a little membrane of its own that's just kind of sitting in there holding on to these glucose transporters, but they're not doing anything because they're internalized. So then when insulin binds, it will cause these to kind of move to the membrane, and then it will fuse with the membrane, and then those transporters will be inserted into the membrane and activated. Okay, and then they can be active. Okay, so that's how this takes place. Again, a problem that has there's a lot of research into what causes type 2 diabetes, and we just don't know all of the things that are going wrong with type 2 diabetes. But one that has been found is that this process isn't taking place. These glucose transporters are not moving to the membrane and being inserted into the cell membrane, so glucose can move through. That's a step that doesn't seem to be happening properly. Okay, so increasing the number of those transporters allows glucose to enter the target cells, which decreases blood glucose. So if you have activation, you put more of these membranes in, or more of these transporters in, and you have more glucose that's moving in, and then that glucose, of course, is turned to glycogen and stored. Okay, so that's a feature specific to glucose that is performed by insulin. So processing and storage of glucose. So let's talk about that, at least the big things. So excess glucose is stored as a complex chain of glucose molecules called glycogen. So this is kind of a nice little sketch of what glycogen is. So glucose are six-sided sugars or ring structures, and it just sticks those together, and it forms these big networks of glucose. So, you know, if you look at things like starches are also, cellulose are also chains of sugar, chains of glucose. So so it's kind of the same thing. Just put it into a chain, and then later, if you need it, you just break glucose off those chains and release it. It's very quick. It's kind of efficient. It's like, here, stick it over here. Okay, we need it. Okay, now, you know, glucagon is here now and telling us to release it. So now we release it. Insulin told us to store it. You know, it's just life in a cell. All right, so when glycogen stores are full, then glucose is converted to fatty acids to form triglycerides. Okay, so now you have excess glucose where you can kind of unfold it, and then you can cut it into its individual carbons, and then you can just stick those carbons into chains, and that is a fatty acid. Okay, this is the most briefest example of fatty acid synthesis, but yeah, you take that chain and then you straighten out that chain, and then you take those 6, and then you just take those and you stick it on, you just bind them, put them together to make a fatty acid. Okay, and that's what you do. Okay, so that's the other thing that insulin does, is it stimulates the production of fats, of triglycerides. Okay, so fatty acids are not the end product. The end product are going to be, in this case, triglycerides. Okay, and that's of course going to happen in fat tissue. So when glucose levels are depleted, okay, so if we don't have enough glucose, then we can have something called, or let's see, when glucose levels are depleted, we have gluconeogenesis, which is the production of glucose. Okay, so this is not done by insulin, so this would be directed by glucagon. Okay, so glucagon is going to produce more glucose. So this is saying that we need more glucose. So we've been throwing this term around. Right now we're just defining it, is really all we're doing. Okay, so the production of new glucose from other molecules such as amino acids, glycerol, or fatty acids. Okay, so one thing that we need to kind of slide in here is the utilization of fatty acids or amino acids as energy. Okay, so the body's favorite form of energy is glucose. Okay, so it's got all the mechanisms involved to turn glucose into ATP, and ATP is what just runs all of these reactions. But it's the glucose coming into the body. That's what we eat to make the ATP. So it likes glucose, and so that's why it has this gluconeogenesis feature, where it can make it from amino acids, glycerol, fatty acids. Okay, so it makes it from a lot of things. However, utilization of fatty acids or amino acids as energy, particularly fatty acids. So in fatty acids, glycerol and amino acids are catabolized to make ATP. They form something called acidic ketones. Okay. Now, ketones can be used as energy, but they are a product of breaking down these fats. So I have this fat up here, and I can break that down to make glucose, to make energy. Okay. So one of the byproducts of that are these ketones. So here's a couple of examples of ketones: acetone, azoacetic acid, and then this one's kind of an important one here, beta-hydroxybutyric acid. Okay. So these are, they call them volatile, and that's because they easily move into the air, and they are aromatic, and so they have a smell. So a lot of times people that are starving will have this fruity smell because of the ketones that are being dissipated, because they're volatile, can move into their breath. Okay. Now, one of the problems with ketones is they are acidic, and they will cause the blood pH to fall. Okay? Low pH means acidic, so less than 7.35. All right, so remember that our ketones are coming from starvation and the inability to, and where you don't have enough glucose, okay? Because we're going to come back and really talk about ketoacidosis later. All right, so the actions of insulin. So the thing to remember is that the overall action of insulin is to increase storage of energy into cells. Okay, so it is a hormone of abundance. We have lots of stuff. We have sugar coming in just everywhere. It's coming in through the intestines, through the walls of the body, and so we have a lot of glucose, and so we have to store that. Okay, so we don't forget, though, that it is other stuff too. Amino acids, fatty acids. Those are also there. Okay, so recall that insulin increases potassium. So potassium also is, insulin will also direct that to be moved into the cells. So yes, we focus on insulin or glucose, but we can't forget that it does all of these other things too. Fat deposition decreases lipolysis. We don't want to break down fat if we're storing fat. Potassium uptake, increased protein synthesis, so it's anabolic, increases protein synthesis inside cells. Yeah, so all of those things. All right, so cell-specific effects of insulin. So what cells are we talking about? I kind of quickly mentioned it earlier, but mainly we're talking about fat cells, so adipose tissue, and skeletal muscle, and liver. Okay, so liver's a big one because that's really its role, is to kind of break down or to store energy and then to release energy, whether it's glucose or fats or whatever, proteins. Liver does that. Skeletal muscle, as we know, has a secondary function, okay, and that is being muscle and helping us walk and stuff. And then adipose tissue is storage of fat. So all of this stuff should make sense. So adipose tissue, well, all of it's going to increase glucose uptake. So glucose uptake is happening in all of them. It doesn't say that here, but glucose uptake. Glucose uptake in the liver too. All right. So in adipose tissue, increases fatty acid uptake. So more fatty acids are moving in. And what happens to fatty acids? They are converted into triglycerides. Okay. Triglycerides for storage. Okay. Remember, fatty acids stuck to a glycerol. That's a triglyceride. Decreases the breakdown. So lipolysis is the breakdown of these fats, and that is not happening. Okay, so it's mostly about fat here. Now, if we have too much glucose, that glucose can be converted into fat too. Okay, as we just saw. Now, skeletal muscle, of course, uptake of glucose. Glycogen storage. So a lot of glycogen storage is going to happen in muscle. That's great. That is fantastic because when we never know when that bear or walrus is going to attack and chase us, and so we might need that glucose to as in an emergency. So yeah, so storage of glucose in glycogen is very important. Increases uptake of amino acids. So one thing that's also true about muscle, it is very high in proteins. So there are enzymes, but there are also structural proteins. So uptake of amino acids and protein synthesis is something that that sort of tracks when it comes to what insulin will do in skeletal muscle. Okay, so it increases that uptake of amino acids. The liver, it really kind of does it all. So increases glycogen synthesis, increases triglyceride synthesis, decreases gluconeogenesis. So remember, insulin is saying we're storing glucose, not making it. Okay, we're storing the glucose we have. We don't need to make new glucose. All right, so it's going to decrease that. And of course, that's going to have an effect on the blood level. All of these things, moving of this glucose in, is going to have the big one, the main one that we monitor, the main one that we look at, the main one that we're concerned about for our patients, and that is it's going to cause a decrease in glucose. But it's also going to decrease fatty acids. It's going to decrease these keto acids. You don't need the keto acids when you have an abundance of nutrients, and it decreases those amino acids that are in the blood. All right, so let's look at some pathology. So diabetes mellitus, it is an abnormality in blood glucose regulation and nutrient storage. So it's related to an absolute or relative deficiency of insulin and or resistance to the actions of insulin. So we've kind of already talked about what diabetes mellitus is in general, and that is when you can't, when you're not able to store glucose, so you end up with that glucose building up in your blood, and then because it's building up in your blood, it's going to move to your urine. It's those glucose transporters in the kidney are going to be saturated, and so you're going to have a buildup of glucose in the urine. All right, and so it's going to be sweet. So mellitus is Latin for sweet, and diabetes is this increase of fluid loss, increase in urine. So you have an increase in sweet urine. Sounds sweet. All right, so essentially the patient can't release insulin or they can't respond to it. So there are a couple of different types here. So both types involve insulin. So in type 1, insulin-dependent diabetes mellitus, it's usually caused by an autoimmune destruction of the beta cells. So remember, the beta cells in the islets, the immune system targets those and kills them and wipes them out. So you don't have a release of insulin. So there is no release, and therefore the cells aren't going to get glucose put into them. It's going to stay in the blood. All right, so then there's type 2 diabetes, and we're going to get into more details of both of these. But that's associated with insulin resistance. So for whatever reason, the insulin may be released from the pancreas, but it isn't working. So the cells are just like, okay, high insulin, yeah, we're not going to do what you said. So that's going to keep the glucose is going to stay in the blood. It's not going to move into the cells. Okay, so high blood glucose, relative lack of insulin. It's not an absolute. Absolute means that you're just not making it. This is relative, which means that there is insulin being made, but it's not having any effect. Okay, so about 90 to 95% of diabetics are type 2, okay, and only 5 to 10% are type 1. Okay. All right, and then there's gestational diabetes associated with pregnancy, also associated with blood glucose, and a lot of pregnant women that have gestational diabetes may go on. They have an increased risk of developing type 2 later on. Okay, and then we already talked a little bit about diabetes insipidus. Diabetes insipidus means flavorless. So their urine is not delicious. It doesn't really taste like anything, apparently. So diabetes insipidus is associated with antidiuretic hormone release. So that is where it's still diabetes. You're still producing lots and lots of urine, but there is not sugar in the urine. Okay, so it's insipidus, flavorless. Okay. All right. So type 1 IDDM, insulin-dependent diabetes mellitus. IDDM, insulin-dependent diabetes mellitus. That is the destruction of pancreatic beta cells, and it prevents the release of insulin. Boom. That's what it is. So what causes it? 95% of the causes are autoimmune, which means the destruction of beta cells. Autoimmune destruction of beta cells. That's 95%. 5% is idiopathic, which means we don't know. I guess we're idiots. We don't know idiopathic, and that's 5%. But so it's kind of just sort of thought of as an autoimmune disease because that's so prevalent. So the thing about an autoimmune, this autoimmune disease, I should say, is that it has a genetic predisposition. Okay? So that means that if a parent is a type 1 diabetic, then their children have an increased risk of having type 1 diabetes. So there's an environmental trigger leads to what's called a T lymphocyte mediated hypersensitivity reaction. Okay, so when we think of T cells, okay, so T cells, those are especially cytotoxic T cells. Cytotoxic T cells will they look at something called MHC. We won't get into too many details, but they look at regular cells that are out here just working away, just doing their thing. That's not the nucleus, that's a smile on their face. And they kind of probe and they say, hey, is there anything wrong in here? And they're sort of looking for little signals out here that may indicate that there's a problem. So let's say that, you know, this is the weirdest discussion of immunology and cytotoxic T cells. But let's say it puts out here and it says on this little, what's called an MHC, and it says, hey, this doesn't look right. Okay, these are all fine. The T cell will be like, okay, well, you know, I don't know what's going on with this one, but this doesn't look right. And it will attack it and it will just kill the cell. So any kind of anomaly, anything that doesn't look right in the cell will cause cytotoxic T cells to kill the cell. That's so in case, okay, if we draw our cell again, in case our cell is over here and our cell is happy, but down over here, there's a virus. Okay, so if there's a virus in there, then pieces of that virus are going to get expressed out here, and that T cell is going to notice that and kill it. I don't know why I'm going into all this detail, but this is why we see a lot of autoimmune disease that are affected by these T cells. Okay, so now beta cells produce the I didn't mean to go into a whole immunology test here, but they produce antibodies. T cells, though, will kill cells, and that's what happens here. Okay, and that's what we've seen before. We see these T cell mediated immune hypersensitivity reactions, and that's what happens with diabetes. Is the T cells will ultimately destroy these beta cells, and so they don't produce insulin. All right, so and we also see T cell mediated. We talked about that before with organ rejection, because they're killing cells. That's what their job is. All right, so it's more common in young people, but it can occur at any age. So usually it's that time period between like 7 and 14, something like that. Let's see, it's an absolute insulin deficiency, so you need insulin replacement, and the type 1 diabetes are prone to ketoacidosis. I want you to remember that, partially because we have it on the next cell, but type 2 is not as prone to ketoacidosis because there is usually still some insulin release and there is still some cell response. Okay. All right, so characterized by increased blood glucose, check. Increased blood fatty acid and keto acid concentration. Okay, we're going to say check on that because we're going to see how that has an effect. Increased blood amino acid concentration. So that's happening because those are supposed to be stored. Fatty acids are supposed to be stored. Amino acids are supposed to be moved into the cell and stored. And then less lean body mass and loss of adipose tissue. So that's just something that's seen. Okay, so let's talk about diabetic ketoacidosis. So it's more common in type 1, as we mentioned. And the reason is because there is a lack of insulin. Okay, so a lack of insulin, so if this is a cell, means that glucose is not going to move into that cell. Okay. Now, remember earlier we said, what is the cells? What is the body's favorite form of energy? And we said it's glucose. Yay. So this cell is not going to get the glucose that it needs to function. So the glucose is here. It's out here. In fact, it's elevated. Okay? So in a type 1 diabetic with diabetic ketoacidosis, their glucose is elevated, but the glucose isn't moving into the cell. Why? Because we don't have insulin. Insulin is the trigger to move that into the cell. So this is a patient that probably stopped taking their insulin. Maybe they ran out, but either way, they're not taking their insulin. Okay, so blood glucose levels are high, but the cells are starving. So glucagon, okay, I'm going to use this picture over here. Is something that release, and that's going to tell you to break down and start using fats as energy. So here's your adipose tissue, so release of fatty acids. And it's like, okay, let's try that. Fatty acids are able to move into the cell, and then inside liver cells, you have the production of ketones. So the breakdown of fatty acids and fatty acid is the breakdown of fats, as we saw earlier, is going to create these ketones or these keto acids. Okay, so I'm trying to give you details without making it too complicated. So glucagon triggers the breakdown of those triglycerides into fatty acids and glycerol, okay, which leads to increased production of ketones in the liver. Okay, so now we have an energy source. Okay, so now we have an energy source of these ketones, but it's also causing the cells to be acidic. Okay, so we're using fats as energy, but we're causing our blood to become acid. So that's what happens. Okay, so that's it. So then we can see the symptoms of that: slow onset, prolonged recovery, history of 1 to 2 days of polyuria because of all this glucose, polydipsia, nausea, vomiting marked by fatigue, may progress to stupor and coma. So this can be deadly because a lot of these symptoms are just going to come from acidic blood. So characteristics: fruity breath, remember, because of those keto acids. Metabolic acidosis, so insulin resistance accompanies severe acidosis. Low dose insulin therapy is indicated with frequent monitoring serum blood glucose. So what does that mean? That means that you give insulin. So you give usually like an IV insulin drip, and then you just provide insulin, and that's how you fix it. I mean, you've got to get that glucose back into the cells, and so they can start functioning again normally without starvation. All right, so compensatory mechanisms: tachycardia. Okay, so tachycardia, you're trying to blow off that carbon dioxide because your blood is acid. Well, your blood's not acid, but it's acidic. And so you're going to have a buildup of carbon dioxide. And so the more you blow off that carbon dioxide, then it shifts that equation that we kept looking at to the left, and your blood glucose or your blood pH will start to move up and out of the acidic range. Okay, so I'm talking about breathing. Tachypnea over here, that's rapid breathing. Tachycardia is rapid heart rate. Okay, so your breathing rate, your ventilation rate will increase to help with the acidosis. All right. So type 2 diabetes, non-insulin dependent diabetes mellitus, characterized by insulin resistance and increased glucose production by the liver. Okay. So your cells are not responding to insulin, and you are actually making more glucose in your liver. So the pancreas produces insulin, but the insulin has little or no effect, and that is the hallmark here of what's going on with type 2 diabetes. So then over time, this happens after many years of either not being compliant or just not being able to be compliant, or just the body doing it. But the beta cells may be damaged, impairing insulin release, possibly requiring insulin injections. Okay, so over 90% of newly diagnosed cases are type 2. Okay, so, and it's also, we already saw that 95% of cases overall are type 2. So usually occurs in people over 30 years of age, but that age is decreasing. It's now, now there are sometimes teenagers who are diagnosed with type 2 diabetes. And it is generally considered progressive, not something that you get better with or you don't recover from. However, non-pharmacological treatments is usually caloric restriction, weight reduction, and that can help. Okay, so that can help reduce the need for medications. Okay, so you still, if you start eating again, if you start, getting into those bad habits of high glucose or high carbohydrate diets again, then you will again start showing the symptoms of type 2 diabetes. So you need to keep that caloric restriction and the weight down. Medications are available to increase pancreas secretion of insulin. One of those is example of those are sulfonylureas. So increase the release of insulin. More insulin will eventually contribute to more glucose being moved into cells and out of the blood. Or drugs that upregulate insulin receptors. More insulin receptors is going to do the same thing. It's going to help move that the glucose into the cell. Okay, and that decrease in insulin resistance. So biguanides, metformin is an example of that. Okay, so let's talk about glucagon and then somatostatin. And then we'll move on to calcium. So glucagon is produced by alpha cells of the islets of Langerhans. It's triggered by decreases in blood concentration, blood glucose concentration. Okay. And so it's kind of doing the opposite of what insulin was doing. So now it's saying blood glucose levels are too low. We need to increase those. So insulin actually decreases the activity of glucagon because when insulin's present, it's saying we're storing energy. When glucose is present, we're saying, hey, we're releasing the energy. We're mobilizing that energy. We're gonna help increase the availability of glucose. So it maintains blood glucose between meals and during periods of fasting. Okay, so sometimes we say that it's a hormone of starvation or something like that, but it's really fasting, low glucose. It's trying to keep it in homeostasis. So this is what we do to raise it. So able to initiate glycogenolysis. So glycogenolysis, remember glycogen. Okay, so glucose will say, oh, time to cut those down and release those individual glucose molecules. Okay, so we can use those to make ATP. So to build new glucose, it stimulates gluconeogenesis to build new glucose from amino acids and fatty acids, increases blood fatty acid and keto acid concentration. So we're breaking down fats now. So glucagon is going to trigger that, which is why if there's no insulin in diabetic ketoacidosis, if there's no insulin to counter that, that's why you have the buildup of ketones. It's because it's saying, hey, break those off, release the fatty acids, and then we're breaking those fatty acids down, and that's going to be that release of ketones. All right, so mobilizes energy. Glycogen to glucose, fats to fatty acids. So this is a fat, these are fatty acids. And then amino acids to form glucose. All right, so factors affecting glucagon secretion. So we need to remember that we're mobilizing energy. Okay, so things that are going to affect glucagon secretion are going to be things like, well, decreased glucose concentration. We have that here, but fasting, higher amino acid concentrations. Those are the kinds of things to remember. So glucagon is the hormone of starvation triggered by decreased nutrients between meals or during fasting, mobilizes energy stores to maintain continuous availability. So the action, we just went through those, breaking down fats and releasing glucose in its purest form. All right, somatostatin. Somatostatin secreted by the delta cells, the islets of Langerhans. So this is pancreatic somatostatin, stimulated by the ingestion of all forms of nutrients, by several GI hormones, by glucagon, by alpha-adrenergic agonists. So basically lots of things that are going to kind of put the brakes on. So that's kind of the role that we've kind of seen of somatostatin with growth hormone, with digestive hormones. It kind of puts the brake on and says, okay, everybody just needs to slow down. So the function specific inhibits secretion of insulin and glucagon, so it kind of controls the output of those within the islets of Langerhans. Secreted in response to a meal, okay? So modulates response to ingestion of food. So the other functions, I just kind of mentioned some that we've talked about. Inhibits growth hormone, inhibits thyroid stimulating hormone, okay? Which is going to kind of cause that, you know, decreases stomach and gut motility. All right, so let's talk about, talking about calcium homeostasis. So calcium homeostasis is important. So homeostasis is so vital that the absence of the hormones for its control would be catastrophic. It's a highly regulated, finely balanced system involving several hormones, and that's what we're going to talk about. We're going to talk about the hormones and the glands that secrete those hormones. So extracellular calcium, remember calcium is in bones. That's what a lot of us, usually if I ask somebody, what's calcium do? It's in bones. But we've already learned that it's vital to muscle contraction. Remember those, the sarcomeres and the cycling, cross-bridge cycling, that involves calcium every step of the way. So it's vital to muscle contraction, including cardiac muscle. So if our calcium levels are low or high, we can have arrhythmias in the heart. And also, we talked about neurotransmitter release from neurons. This is a good review. And many other structural and physiologic functions that we've kind of seen along the way. So hypocalcemia is low or decrease in extracellular calcium. So there is hyperreflexia. Now, I want to talk about that. I'm just going to draw a cell here. It could be a muscle cell. It could be a nerve cell. It doesn't look like either one. But if you have, remember the cell is negative on the inside. Okay? Now, we already talked about how much sodium is on the outside. Okay, and and we have, let's see, so we have a lot of sodium on the outside, and we don't have very much calcium on the inside. So most of the calcium on the inside is kind of sequestered in the sarcoplasmic reticulum or the endoplasmic reticulum. So, and we have high potassium on the inside, and then we have a bunch of negative ions on the inside of the cell. So overall, we have a negative charge on the inside of the cell. So the reason I'm talking about this is because calcium, even though its concentrations aren't very much, are found more on the outside of the cell. So that means that's contributing to this positive on the outside. Okay, so calcium is contributing to that. So during hypocalcemia, it's not as positive. Okay, which means that, I'm going to just erase them all, which means that this polarization is not as polarized. So that means if, let's say if this was going to be minus 70, when we had all these positives, when we get rid of that calcium levels are low. Of course, it's not going to be gone completely or you die, but it might be down to minus 60. Okay, just because these positives are gone now. So it kind of drops down to minus 60, which means that it's going to be more sensitive. More sensitive, which means a trigger that's going to cause your heart or your muscles to go, nerve conduction to go. Anything that's going to depend on this polarization is going to happen more quickly. Okay, so hypocalcemia, hyperreflexia. Reflexes are going to be hyper, they're going to be jumpy, twitching, muscle cramps, tingling numbness. That's going to affect the nerve conduction as well. And then we have a couple of things down here. I'm not going to really go into them. Shavstek sign, twitching of facial muscles. So that's a twitching kind of a reaction when tapping the facial nerve. Trousseau sign, hand spasms when you kind of reduce blood flow to the hand by pumping up a blood pressure cuff. So these things are kind of all along that hyperreflexia kind of response. Hypercalcemia is going to be the opposite. Too much calcium, that means you're increasing the positive, okay, which is going to make it, positive, which is going to make it harder to depolarize the cell. So now, because you've got more positive on the outside, this might be minus 80 on the inside. And that's going to make it harder. So it's going to hyporeflexia, lethargy, constipation, because you aren't going to have peristaltic movements because of that hyporeflexia. Polyuria, because you've got more calcium that's moving through the nephron, the tubules. Polydipsia, thirst, and then just coma and death after that. Okay, so how's calcium carried in the blood? So calcium is carried in the blood, but about 40% is actually bound to plasma proteins like albumin. 60% is filterable, and when we say filterable, we mean filterable in the nephron, in the tubules. So it can move through this is the glomerulus here. Okay, so that means that calcium, about 60% can move through. But remember, 10% is bound to anions, particularly phosphate. Okay, so we had those phosphate anions that calcium can bind to. Okay, But those can be filtered, and that's part of our urine buffer, is those phosphates, okay? So, but overall, what we end up with is we have about 50% in our blood that is just free ionized calcium 2+. Okay? So that's about half. All right, so factors that affect blood calcium. So these plasma protein concentrations. So increased blood proteins will increase total calcium. Okay. Because it's going to bind more of the calcium, and if that happens, if that's kind of a normal thing, then you may not really notice a difference in blood calcium levels, free calcium, but you will have more calcium just because more will be bound. Plasma ion concentration. So if you have more, now this will have an effect. So if you have more phosphate ions in the blood, then that will tie up those calcium. So if you have the phosphate and then those calcium are like looking at it like, hey, you know, I'm gonna 2+, I'm gonna bind to that. Well, that's not free calcium. Okay, so that's going to decrease that free calcium. And then acid-base abnormalities. So there's a nice picture over here that shows what happens is that more hydrogen. So hydrogen and calcium, so we can think of this as hydrogen and calcium are competing for binding sites on albumin, okay, or on plasma proteins. And so when you have acidemia, then you're going to boot off, the hydrogens are going to kick off some of the calcium and say, hey, we're going to bind to the albumin now because we're in charge, because we have acidic blood. Okay, and that's just going to increase your calcium that's out here. So the calcium is going to be sad, but it's also going to be out in the blood. So I don't know, maybe it's happy, I don't know. But then alkalemia is the opposite. So if you don't have that that hydrogen competing for binding sites, then you're going to have decreased calcium. So you're not going to have as much calcium out here. Okay, so the thing to remember is that when you have a patient and you're looking at their labs and they are acidotic, you can kind of predict that they may have higher ionized calcium, and may have to do something about that, because high calcium can, you know, cause, as we've seen, problems, and so can low calcium for alkalemia. Okay, so the control of calcium homeostasis. How do we control it? There are 3 different ways. So the bone is how we're storing calcium. Okay, it's stored conveniently enough along with phosphates. Okay, and something called hydroxyapatite. We'll talk about that in a minute. But that's how calcium is stored. It's stored in the bone, and there's plenty of bone. But if your calcium levels are low and you start stealing calcium from your bones are going to get weak. The kidney. So kidney is, as we usually think of it, well, the kidney can control sodium, so why not calcium? And it does. It controls the reuptake of calcium. Okay, so reabsorption of calcium can happen by the kidney, and it can also get rid of phosphates. Kidney can do lots of things. And that also just happens to be where vitamin D is activated. And we'll talk about what vitamin D does. But vitamin D has its effects in the intestines. I guess we could talk about it now. It causes absorption of calcium in your diet from the intestines. But that's activated vitamin D that does that. Okay, so the 3 hormones that are involved that we're going to talk about are parathyroid hormone. Parathyroid hormone sits on their little tiny glands that sit on the thyroid gland. Okay, so those parathyroid hormone, or the parathyroid gland sits on the thyroid gland. The parathyroid hormone is the hormone that the parathyroid gland produces. Okay, so increases, this is what PTH, parathyroid hormone, does, increases reabsorption or resorption from the bone, so it's going to weaken your bones if it's active too long. Otherwise, in a healthy person, it's just gonna cause some cycling there. So increases resorption from the bone and reabsorption from the kidney. So it's going to steal it from the bones, and it's going to keep the kidneys from peeing it out. So you're going to reabsorb it in the kidneys. So overall, it's going to increase your calcium levels. It also directs the activation of vitamin D in the kidney. So we mentioned that vitamin D is activated in the kidney. Vitamin D. And vitamin D is a hormone. Activated vitamin D is a hormone that has its effect in the intestines, which is going to allow you to absorb more calcium. So vitamin D increases calcium. Okay. So the active form of vitamin D increases calcium. It's just activated in the kidney. So let's see. So the active form of vitamin D, we just said what it does. It increases bone resorption. Let's see. Increases bone resorption inhibited by calcitonin. So yes, I didn't mention that. Let's see. But yeah, bone resorption here. Okay, and absorption in the kidney. So increases bone resorption in the bones. It's inhibited by calcitonin, which we'll also talk about, and increases absorption in the intestines. Trying to keep all my absorption terms correct here. We could probably just use the same one for all of them, but they technically mean slightly different things. All right, so let's look at the parathyroid glands and the parathyroid hormone. So here are your parathyroid glands. Like I said, they're just tiny little glands that sit on the back of the thyroid gland. Okay, so this is posterior. And they secrete parathyroid hormone. Okay. So 4 small glands located in the neck on the posterior part of the thyroid gland. Parathyroid hormone is critically important for regulation of calcium levels. It is going to increase calcium levels. Remember that. And we also have to remember how. So when calcium concentration decreases, PTH is secreted by the parathyroid glands. Yes, I mean, that makes sense. And parathyroid has its effects on the bone. Remember, resorption of calcium from the bone, and the kidney. And in the kidney, it is causing reabsorption of calcium, and it's also getting rid of the phosphate that was tying up the calcium in the blood. And it has an effect on the activating vitamin D to increase calcium absorption. So it has its effects on the intestines. So parathyroid hormone is an 84 amino acid polypeptide, so it's a polypeptide hormone. So secretion varies to help maintain calcium levels. We've seen all of this. I mean, it makes sense that it would vary from maximum release to a very minimal release. So it varies, but it only varies within this range here. So calcium sensing receptors on parathyroid membranes, so this is in the parathyroid gland, monitor this extracellular calcium. So the presence of calcium is going to prevent the release of PTH because there's calcium there. It doesn't need to be released. But when calcium levels are low, so if our calcium levels are low down here, this is low calcium, so calcium is on our x-axis here. If our calcium levels are low, then we have maximal release of PTH. When our calcium levels are high, over here, then we have minimal release. And so it kind of works in this range. And really, the range is about 8.5, normal range, to about 10.5 or 8.3 to 10.3. Different hospitals use different ranges, but it's all about that, which is kind of in this range here. So this is where your parathyroid hormone release will kind of be altered. Okay, so that kind of fits. Okay, so the actions of it. What is parathyroid hormone doing? In the bone, it is primarily made of calcium phosphate. Let's see. Okay, so in the bone, gosh, I think I'm rushing. Bone is primarily made of calcium phosphate lattice structure called hydroxyapatite. So it's calcium phosphate, so calcium bound to phosphate. And this is what makes up the bony matrix, okay? So that's what makes bone, okay? So within that bone, we have osteoblasts. So osteoblasts are responsible for bone formation. And then we also have osteoclasts, and osteoclasts are a little bit bigger. And osteoclasts are responsible for breakdown. And then we just have our regular osteocytes, which are just bone secreting cells. So we have those spread around there too. All right, so that's kind of a terrible histological representation of a slice of bone. But what we need to remember is that we have osteoblasts. Blast, usually when you see blast, you're thinking building up, producing, blast, are forming bone. Clasts are responsible for breaking down bone. So now we have our mechanism for how are we breaking down and stealing calcium from bone, and then how are we building that bone back up. So we know we have individual cells that are responsible for that. So parathyroid hormone ultimately increases osteoclast activity. The reason I put the word in ultimately there is because it does it in some steps that also involve osteoblast. But it's remodeling and will ultimately break down the bone and resorb or steal calcium and also phosphate from the bone. But it breaks down this hydroxyapatite so it can get the calcium back in the blood. Okay, so and this is going to be osteoclast activity that's ultimately doing that. Okay, so the orange one up here is breaking it down. Osteoclast. So in the kidney, parathyroid hormone has two actions. So I've been kind of alluding to this, but it stimulates calcium reabsorption. I think that's pretty easy to remember. So if we have our renal tubule here, it's saying that calcium is being reabsorbed. Okay, we like that, calcium to be reabsorbed. However, it inhibits phosphate reabsorption. So it's going to say, hey, calcium, how you doing? And it's going to tell phosphate, yeah, you can just keep going. And so phosphate will leave as urine, in the urine. Okay, so by inhibiting sodium phosphate co-transport, by excreting phosphate, oh yeah, that's how it does it. But by excreting phosphate, calcium remains unbound in the plasma. So remember, that was kind of an issue, is we had calcium bound to phosphate. Okay. Well, now we're saying bye-bye phosphate. So now calcium, we're going to have more free calcium. Okay. So that's good. So if you have hypokalemia, if your calcium levels are low, that's a good response, is increasing that calcium. Okay. So in the small intestine, we've mentioned this indirect effect by activating vitamin D. So in the kidneys, PTH stimulates the renal enzyme to convert 25-hydroxycholecalciferol to the active form, which is 1,25-dihydrocholecalciferol. I like to just say inactive vitamin D and activated vitamin D. So activated vitamin D, which is this one, the 1,25-dihydrocholecalciferol, if you prefer that. But activated vitamin D stimulates intestinal calcium absorption. Okay? So everything is right there. Resorption from the bone, let's get it back into the blood. Let's get rid of the phosphate that's been tying it up. So now we have more free calcium, and let's just bring in more from the diet. Okay? Let's absorb all we can. All right. So, regulating parathyroid hormone secretion and actions on the bone, kidney, and the intestines. So, obviously, this is going to be a feedback mechanism. So, low plasma calcium is going to increase parathyroid hormone secretion, and then it has these effects. So, I like this graph because it kind of just shows you right there when it seems like, oh, it does so much. It does 1, 2, 3, 4, 5 things, and really 4 that we care about. So bone resorption, yep, that's those osteoclasts. Okay, so we do have to remember some details about this. The kidney decrease phosphate reabsorption, so get rid of the phosphates and increase the calcium reabsorption. And I'm not really counting this one, urinary cyclic AMP. So you're going to have an increase in urinary cyclic AMP just because the process of blocking phosphate reabsorption is going to produce cyclic AMP. Okay. and that's going to end up in the urine. Okay, so not too concerned about that, because it's just part of this process of saying, hey, phosphate, you need to leave, okay, because the calcium just likes you too much. All right, and then the intestine, increased calcium absorption by activated vitamin D. Okay, so hopefully that makes sense. Okay, so pathophysiology of parathyroid hormone. Well, if your parathyroid is too active, you're going to have too much calcium. Okay, so that shouldn't be a stretch to come up with that. But that's true. So hyperparathyroidism, hypercalcemia due to increased bone resorption and renal reabsorption. Hypophosphatemia due to increased excretion. So now we don't have enough phosphate. So this is a pathological situation, a pathology situation, where things are out of control and going the wrong direction because your parathyroid hormone is either gone or is not functioning. Okay. Constipation from reduced intestinal motility, okay, because you have too much calcium, it's harder to depolarize cells for that motility. Increased risk of osteoporosis because you're stealing all the calcium away from the bones. Okay, so maybe treated. Wait, I think I said that you didn't have a parathyroid. Yeah, your parathyroid is too active. It's too active in hyperparathyroidism. Okay, so maybe treated then, okay, by surgical removal of parathyroid glands. I think I said something wrong, but I'm sure I did. Okay, so so let's go the other direction. So, yep, we have to make these make sense. Okay, remember that. And then hypoparathyroidism, usually a consequence of thyroid surgery. Okay, so this is kind of common in people with thyroid surgery. Because it's on the thyroid, it can a lot of times be also removed, despite efforts not to. So, or cancer treatments can also do it. So for, so thyroid surgery for hyperthyroidism. So if somebody has hyperthyroidism or a cancer or something like that, it has to be removed. Or parathyroids can be removed themselves. So hypocalcemia, okay, from decreased bone resorption and renal reabsorption. So remember, our parathyroid is trying to increase calcium, and now our parathyroid hormone is low. So we're going to have more phosphate tying up our calcium. We're going to have decreased calcium being reabsorbed, and you're also not going to have, but you can treat it with active vitamin D and calcium. Okay, so just provide calcium to keep the calcium levels up, but you also need vitamin D to increase that absorption. Okay, it depends on how severe it is, how it's treated. Okay, so a couple more that we've been talking about. We haven't really talked about calcitonin at all. It's kind of forgotten. But calcitonin the primary action of calcitonin is to inhibit osteoclast-mediated bone resorption. Remember, the osteoclasts were causing that reabsorption of calcium, which is going to decrease plasma calcium levels. So it's kind of offsetting the vitamin D, which was causing that resorption. And this is saying, no, we need the calcium in the bone, so we're going to just leave it there. So that's the role of calcitonin. So it's a 32 amino acid polypeptide released by cells of the thyroid gland. Okay, thyroid gland, not parathyroid. Okay, and it wants to maintain or increase calcium levels. Okay. That's important. So release is stimulated by increased plasma calcium. Let's see, inhibit osteoclast media decreasing. Yes, right. So, okay, I said that wrong. It doesn't want to increase calcium level. It wants to increase bone calcium. That's what I knew there was something wrong with how I was saying that. So we're preventing the bone from breaking down, which means that we're keeping the calcium in the bone. Yes. Okay. I say that because a lot of times calcitonin is like a treatment for like osteoporosis, things like that, to prevent bone breakdown. So we want to keep that calcium in the bone. So release is stimulated by increased plasma calcium. So if blood calcium levels are high, then we can, again, increase the bone calcium, keep it stored in the bone. It does not have, and this is one of the reasons why it's kind of not really forgotten, but it doesn't seem to play a super critical role. It does not have a major impact on minute-to-minute calcium homeostasis, so we know it does have a role. It does increase it does maintain the bone structure, but it's not thyroidectomy, thyroid tumors. Remember, calcitonin is being released by the thyroid gland. It doesn't have a significant impact on calcium regulation. So we're not sure what the role of calcium regulation is. We do know that it's functioning to maintain it in the bones. Okay. So vitamin D. So vitamin D is a vitamin that's actually considered a hormone because it moves through the blood and it has its effect at a distant place. So the role is to increase plasma concentration of both calcium and phosphate. Okay. So remember, this is important and easy to get lost with, but remember PTH, parathyroid hormone, increased calcium but decreased phosphate, PO4, whatever. Okay. So yeah, So remember, parathyroid hormone did that. This is going to increase both calcium and phosphate. So again, where we were talking about calcitonin and we were kind of saying, yeah, but it kind of builds up the bones. So does this. Okay, so this is the focus on building up bones. All right. So because you're putting calcium and phosphate together, remember calcium and phosphate were part of that hydroxyapatite, which is part of the structure of the bones. Okay, it's that lattice network of calcium and phosphate. Okay, so vitamin D is good for you for that reason. So it's provided in the diet and manufactured in the skin from cholesterol. So UV light can manufacture vitamin D. So the vitamin D is cholecalciferol. That is inactive as we get it in our diet and as we get it from the sun. And it is activated in the kidney to 1,25-dihydrocholecalciferol. I know I've mentioned that before, but that's it. This is the structure of it here. So it's ultimately from cholesterol. Okay. All right. Let's see. Yeah. 7-dehydrocholesterol, yeah, and then it's manufactured. So activation occurs in that kidney by the renal enzyme 1-alpha hydroxylase, and that is going to, that's going to cause the activation of it, and then it's active. So so activation is going to be caused by low calcium levels. Increased parathyroid hormone is going to also activate that. We saw that before. And decreased phosphate levels will also increase the vitamin D activation. Okay, so what is it doing? Actions of vitamin D, increase calcium and phosphate absorption. So the mechanism, so this is in the intestine, okay? The mechanism is from production of calcium binding protein, something called calbindin, that can bind 4 calciums and then release them into the bloodstream. So what are we talking about? We're saying that here is the calcium that's in the lumen of the stomach, and it can move into the cell, okay? But it doesn't move into the cell rapidly, or it doesn't move across the cell rapidly. So we have a binding protein that will pull it in, keep the free calcium levels low because it's bound, and then it will release that into a calcium pump to release it into the blood. Okay, so it's just this mechanism to move calcium in, and that's really, we don't necessarily need to know exactly details of that mechanism, but that's what vitamin D is doing, is causing that to happen. In the kidney, it stimulates calcium and phosphate reabsorption. So we mentioned that already. And then we also mentioned that this difference in phosphate, because it's phosphate and calcium. And the bone stimulates remodeling and formation of new bone. Okay, so like I said, vitamin D is important for bones. So stimulates osteoclasts to promote the resorption of old bone. So remember, so osteoclasts are breaking down, and so that's what I say, these mechanisms will activate both osteoclasts and osteoblasts, but overall, so we break down the old bone and we build up new bone, okay, and that's really the ultimate effect, okay. The phosphate and calcium are then used from those broken down osteocytes to build it back up again. So it's like tearing down a highway and then building a better one. That's the idea. Okay, or a bridge. All right, so pathophysiology of vitamin D. Rickets is a vitamin D deficiency in children. So insufficient calcium and phosphate are available for growing bones, so the bones do not grow properly. So manifests as a growth failure and skeletal deformities. Okay, bow-leggedness and difficulty, just the bones are not growing properly. Osteomalacia is vitamin D deficiency in adults. Okay, so also weakened bones and fragile. So vitamin D resistance, the kidney is unable to produce the active form of vitamin D. This one, I think, warrants some attention, the kidney. So this is something that happens with chronic kidney disease patients, okay, which you'll see. So chronic kidney disease patients are often prescribed the active form of vitamin D. Remember that, because they can't, it's like, oh, they don't, I mean, if your thinking is they don't have vitamin D, so we're going to give them vitamin D or tell them to stay out in the sun a little more, that's not going to work, because the problem is not vitamin D, the problem is the active form of vitamin D that's supposed to be produced in the kidney and is not. Okay, so remember that. All right, osteoporosis. Not sure why that's here, because it has really nothing to do with calcium. But estrogen, it's in your book though, and it's worth talking about. So estrogen and testosterone are needed for maintenance of bone mass. So in older age, declines in testosterone and estrogen is a risk factor for osteoporosis. Particularly, we see that with estrogen decline after menopause. Okay. All right, this was super long, and I apologize, but it's done.